Hello everyone, and welcome to our study session. Today we're diving into fatty acid oxidation, part II. This is part of Module 2. I'm glad you're here, and I look forward to exploring this essential topic with you. In this session, we'll cover key aspects of fatty acid metabolism, focusing on the processes and mechanisms involved. So let's get started and deepen our understanding together. Welcome to today's session. In this part, we'll explore the main content of our topic, starting with the oxidation of unsaturated fatty acids. We'll look at how these fatty acids are broken down, which is crucial because they are a major energy source. Next, we'll examine the oxidation of fatty acids that have an odd number of carbons, a slightly different process that requires special enzymes. Then, we'll discuss alpha and omega oxidation pathways, which are alternative routes for fatty acid degradation, especially when the main pathways are impaired. Moving on, we'll cover how the body regulates fatty acid oxidation to maintain energy balance and prevent metabolic issues. We'll also dive into ketone bodies, which are important alternative fuels, particularly during fasting or low carbohydrate intake. After that, we'll consider some clinical aspects related to fatty acid metabolism, linking theory to real-world health conditions. Following this, we'll look at tutor-marked assignments, whether individually or in groups, to reinforce learning. Finally, we'll wrap up with a summary of key points and some self-assessment questions to test your understanding. Throughout this section, I'll emphasize how these processes connect to overall metabolic health and energy management in the body. Although beta-oxidation is the main pathway for breaking down fatty acids, it's important to remember that it primarily targets even-numbered saturated fatty acids. When we deal with other types of fatty acids, such as unsaturated ones or those with an odd number of carbons, the process needs some modifications. These modifications ensure that these less common fatty acids can still be efficiently broken down and utilized for energy. In addition, animal tissues have minor pathways that involve oxidation at the alpha and omega carbons. These pathways are not the primary routes, but play important roles in metabolizing specific fatty acids. The products generated from alpha and omega oxidation can then feed into the main beta-oxidation pathway, ensuring maximum energy extraction. Now, during fasting or starvation, or in conditions like diabetes mellitus, where the body can't efficiently use glucose, ketone bodies become crucial. They serve as alternative fuel sources, especially for the brain and muscles, helping to maintain energy levels when glucose is scarce. Lastly, it's worth noting that there are several disorders related to fatty acid oxidation. These include conditions like carnitine deficiency, Rifsum's disease, Jamaican vomiting sickness, Zellweger's disease, and ketoacidosis. Recognizing these helps us understand the importance of proper fatty acid metabolism for overall health and disease prevention. Most fatty acids, especially saturated ones, can be broken down through the beta-oxidation pathway, which we discussed in the previous slide. This process is quite efficient for most common fatty acids. However, there are some exceptions. Fatty acids that contain an odd number of carbon atoms, certain types of unsaturated fatty acids, and methylated fatty acids require some modifications to the standard beta-oxidation sequence. These modifications are necessary because their structural features can interfere with the normal breakdown process. Understanding these alternative pathways and modifications is important because it highlights the diversity and complexity of fatty acid oxidation. It also helps us appreciate how the body adapts to different types of fatty acids to meet energy needs. So, in this slide, we're exploring these other mechanisms of fatty acid oxidation that go beyond the basic pathway. After studying this study session, I expect you to be able to describe the pathways for oxidation of unsaturated fatty acids and fatty acids with an odd number of carbon atoms. I want you to understand how these pathways differ from those of saturated fatty acids and why they are important. Next, you'll be able to describe alpha and omega oxidation of fatty acids, which are alternative processes that come into play when the usual beta-oxidation is impaired or defective. We'll also cover how fatty acid oxidation is regulated, so you can grasp the key control points that determine when and how these processes occur in the body. Additionally, you'll learn how ketone bodies are utilized for energy production, especially during periods of fasting or carbohydrate restriction, which is a crucial adaptive mechanism. Finally, we'll discuss some disorders of fatty acid oxidation, highlighting how disruptions in these pathways can lead to clinical conditions. Connecting all these points helps us see the bigger picture of energy metabolism and its significance for health and disease. When we look at the first step of beta-oxidation, the double bond formed between the alpha and beta carbons is in the trans configuration. However, most unsaturated fatty acids naturally have cis double bonds, which are different in structure. When these cis double bonds are hydrated by the enzyme hydratase, they typically produce D-isomers. These D-isomers can't continue directly through beta-oxidation, so they are converted into L-isomers by an enzyme called racemase. Once converted, beta-oxidation can proceed normally. Now, there's an important exception to keep in mind. If the double bond in an unsaturated fatty acid is located between the beta and gamma carbons, not between alpha and beta, the fatty acid can't enter beta-oxidation directly. In this case, an enzyme called isomerase steps in to move that double bond to the correct position between alpha and beta so that oxidation can continue smoothly. This process highlights how specialized enzymes help us handle different structures of unsaturated fats, ensuring efficient energy extraction from a variety of fatty acids. Let's take a look at how fatty acids with an odd number of carbons are broken down. During beta-oxidation, these fatty acids are progressively shortened by removing 2 carbon units at a time. This process continues until we're left with a 3-carbon molecule called propionyl-CoA now. This is a key point because, unlike even-chain fatty acids, which enter the TCA cycle directly as acetyl-CoA, odd-chain fatty acids produce this propionyl-CoA, which needs to be converted into a form that can enter the cycle. So what happens next? Propionyl-CoA is first carboxylated by an enzyme called propionyl-CoA carboxylase. This reaction adds a carbon dioxide molecule, converting it into dimethylmalonyl-CoA. Then, a specific enzyme called methylmalonyl-CoA racemase takes over, converting the D-isomer into the L-isomer, which is the form that can be further processed. Finally, the L-methylmalonyl-CoA undergoes a rearrangement catalyzed by methylmalonyl-CoA mutase. This step transforms it into succinyl-CoA, a molecule that can directly enter the TCA cycle. Once inside the cycle, succinyl-CoA can be oxidized further to produce carbon dioxide and water, releasing energy, or it can serve as a precursor for gluconeogenesis, helping to maintain blood glucose levels. So, in summary, the oxidation of odd-chain fatty acids involves a series of conversions that ultimately feed into the TCA cycle, highlighting the body's versatile mechanisms for energy production and metabolic flexibility. Let's take a closer look at the oxidation of fatty acids that have an odd number of carbons. Unlike the more common even-numbered fatty acids, these require a slightly different approach during their breakdown. As you may recall, beta-oxidation typically cleaves 2 carbon units in the form of acetyl-CoA, but when dealing with odd-chain fatty acids, the process leaves us with a 3-carbon fragment called propionyl-CoA. This is a key distinction. Now, this propionyl-CoA can't directly enter the citric acid cycle, so it first needs to be converted into a more useful form. The body converts propionyl-CoA into succinyl-CoA through a series of reactions involving vitamin B12-dependent enzymes. This succinyl-CoA then feeds directly into the citric acid cycle, allowing energy extraction to continue efficiently. This pathway highlights how our metabolism is adaptable, ensuring that even odd-chain fatty acids can be utilized for energy. It's a perfect example of the interconnectedness of different metabolic pathways, reinforcing the theme of metabolic flexibility that we've been exploring throughout this presentation. Animal tissues contain minor pathways that involve oxidation of fatty acids at the alpha and omega carbons. These pathways are less prominent than the main beta-oxidation process, but still play important roles in specific metabolic contexts. The products generated from alpha and omega oxidation can actually enter the beta-oxidation pathway, contributing to energy production. Specifically, alpha-oxidation occurs near the carboxyl end of the fatty acid, while omega-oxidation happens at the methyl end. These processes help in breaking down fatty acids that might be resistant to beta-oxidation or are already partially oxidized. Once these oxidation products form, they can be funneled into beta-oxidation, ensuring the fatty acid molecules are fully utilized for energy. This highlights the interconnectedness of fatty acid metabolic pathways and their flexibility in meeting the body's energy needs, especially when typical pathways are overwhelmed or impaired. So, even though alpha and omega oxidation are minor routes, they are crucial backup systems that help maintain efficient fatty acid metabolism, particularly in certain tissues or under specific metabolic conditions. Today, I want to talk about a fascinating aspect of fatty acid metabolism called α-oxidation. During their metabolism, some fatty acids undergo a process called hydroxylation, specifically on the C2 position, which is also known as the alpha carbon. This means we add a hydroxyl group there, transforming the fatty acid into an alpha-hydroxy derivative. What's particularly interesting is that these alpha-hydroxy derivatives can be further oxidized, leading to the release of carbon dioxide and the formation of a fatty acid that has one less carbon atom. This shortened fatty acid can then enter the beta-oxidation pathway, continuing its breakdown for energy. Alpha-oxidation primarily takes place in the endoplasmic reticulum, and it's especially important when dealing with methylated fatty acids. These methyl groups can make beta-oxidation tricky, so alpha-oxidation provides an alternative route. In fact, alpha-oxidation is a key method for generating odd-chain fatty acids, which are less common but play important roles in metabolism. So, in summary, alpha-oxidation helps us handle methylated fatty acids and odd-chain fatty acids by modifying their structure, allowing them to be efficiently broken down and utilized for energy. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about fatty acids that are hydroxylated on the terminal carbon. These can undergo a process called omega-oxidation. Essentially, the hydroxyl group at the omega position gets converted into a carboxy group, which transforms the molecule into an alpha-omega dicarboxylic fatty acid. Now, this is interesting because once you have this dicarboxylic acid, it can enter the beta-oxidation pathway from both ends. This dual entry point allows for more versatile processing of the fatty acid. It's also worth noting that omega oxidation primarily occurs in the endoplasmic reticulum of liver cells. This pathway becomes especially relevant when other metabolic pathways are overwhelmed or impaired. Understanding omega oxidation helps us appreciate how the body handles fatty acids in different contexts, contributing to overall lipid metabolism and energy production. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about how fatty acid oxidation in the mitochondria is regulated. The key factor here is controlling how substrates enter the organelle. The main enzyme involved in this process is CPT1. During the fed state, malonyl-CoA, which is produced during fatty acid synthesis, actually inhibits CPT1. This makes sense because when you're eating and synthesizing fats, you don't want to simultaneously break down fatty acids. Conversely, in the fasted state, CPT1 becomes very active, promoting fatty acid oxidation to meet the body's energy needs. Interestingly, fatty acid oxidation in muscle is also regulated by malonyl-CoA, even though muscle tissue doesn't synthesize fatty acids itself. Instead, muscle contains an isozyme of acetyl-CoA carboxylase that produces malonyl-CoA solely to regulate CPT1 activity. This enzyme's activity is influenced by citrate, which activates it, and by phosphorylation, which inhibits it. Phosphorylation of this enzyme is mediated by both protein kinase A and AMP-dependent kinase. When protein kinase phosphorylates it, fatty acid oxidation can be regulated based on dietary status. For example, in the fed state, high insulin levels lead to low phosphorylation, allowing fatty acid oxidation to proceed. Overall, this regulation ensures that fatty acid oxidation is finely tuned according to the body's energy and nutritional state. Now, the enzyme we're focusing on produces malonyl-CoA, which plays a crucial role in regulating fatty acid oxidation. It does this by inhibiting CPT1, effectively blocking the process. There's also a second kinase involved here, and it's regulated by AMP levels. This kinase essentially links the rate of fatty acid oxidation to the muscle's energy status. When the muscle is at rest, AMP levels are low. Because of this, the AMP-dependent kinase remains inactive. As a result, acetyl-CoA carboxylase stays active, leading to the production of malonyl-CoA. This malonyl-CoA then inhibits CPT1, creating a feedback loop that controls fatty acid entry into the mitochondria. But when the muscle needs energy, the situation changes. The inhibition of acetyl-CoA carboxylase is lifted, leading to lower malonyl-CoA levels. With less malonyl-CoA, CPT1 is no longer inhibited, which activates fatty acid oxidation. So overall, this mechanism tightly links the muscle's energy demands to the regulation of fatty acid oxidation, ensuring efficient energy use. Ketone bodies are water-soluble products of lipid oxidation that are formed in the liver and kidney mitochondria during prolonged fasting. These molecules, mainly acetoacetic acid and its reduction product, hydroxybutyric acid, are made from acetyl-CoA now. This acetyl-CoA is produced during the breakdown of fatty acids and amino acids. The synthesis of ketone bodies occurs specifically in the mitochondrial matrix. It all starts with the condensation of two acetyl-CoA molecules to form acetoacetyl-CoA. Interestingly, this reaction is actually the reverse of the final step in β-oxidation, which is quite a fascinating aspect of their chemistry. The enzyme responsible for this step is beta-catechiolase, and it's actually an isozyme of the enzyme involved in beta-oxidation. After that, HMG-CoA synthase comes into play, catalyzing the condensation of acetoacetyl-CoA with another acetyl-CoA to produce HMG-CoA, or beta-hydroxybeta-methylglutaryl-CoA. Next, HMG-CoA lyase cleaves HMG-CoA to generate acetoacetic acid and another molecule of acetyl-CoA. Some of this acetoacetic acid is then reduced to d-beta-hydroxybutyrate by the enzyme beta-hydroxybutyrate dehydrogenase, which takes place in the liver mitochondria. The extent to which this reduction occurs depends on the NADH to NAD+ ratio inside the mitochondria. During fasting, for example, fatty acid oxidation produces NADH, shifting the balance and promoting the formation of hydroxybutyrate. So, in summary, ketone bodies serve as an important alternative energy source during prolonged fasting or carbohydrate restriction, and their synthesis is tightly regulated by mitochondrial enzyme activity and the redox state. Part of this process involves the reduction of acetoacetate, which is a key ketone body. The beta-hydroxybutyrate and acetoacetate are released from the liver and kidney, becoming available for use by other tissues that need energy, especially during fasting or carbohydrate restriction. Some acetoacetate also undergoes a slow, spontaneous, non-enzymatic decarboxylation to produce acetone. Under normal conditions, this acetone formation is negligible, but in cases of severe diabetic ketoacidosis, where acetoacetate levels are very high, acetone can accumulate enough to be detectable in the breath. Now, I want to highlight that HMG-CoA is not only involved in ketone body formation, but also plays a crucial role in cholesterol synthesis. Interestingly, the HMG-CoA used in these two pathways, ketogenesis and cholesterol synthesis, are kept in separate metabolic pools. The HMG-CoA for ketogenesis is produced specifically in hepatic mitochondria by an isozyme of HMG-CoA synthase that is highly active during prolonged fasting. Furthermore, the enzyme HMG-CoA lyase, which converts HMG-CoA into acetoacetate and acetyl-CoA, is only expressed in hepatic mitochondria. This compartmentalization ensures that different processes, ketone body production and cholesterol synthesis, are tightly regulated and do not interfere with each other. This distinction is vital for understanding how our body manages its energy sources efficiently, especially during states of fasting or metabolic stress. In contrast to the main pathway, HMG-CoA for cholesterol synthesis is actually produced at low levels in the cytosol of many tissues, thanks to a cytosylic isozyme of HMG-CoA synthase. Now, moving to ketone bodies, acetoacetate and beta-hydroxybutyrate, they are excellent fuels for many tissues outside the liver, such as the heart, skeletal muscle, and brain. This is especially true when glucose is scarce or not efficiently used. In fact, the brain begins to utilize ketone bodies after about 2 to 3 days of fasting. This shift helps reduce the brain's dependence on glucose, which in turn decreases the need for gluconeogenesis. That is, the production of new glucose during prolonged fasting periods. This process actually helps spare muscle proteins, which is quite beneficial. Additionally, ketone bodies serve as precursors for the synthesis of lipids in the brain during the neonatal period. When they're used as fuel, they're metabolized in the mitochondria of non-hepatic tissues. For example, beta-hydroxybutyrate is converted to acetoacetate by an enzyme called beta-hydroxybutyrate dehydrogenase, which uses NAD as a cofactor for oxidation. Finally, acetoacetate itself is converted into its CoA derivative by an enzyme called acetoacetate succinyl-CoA transferase, which is present in various tissues. This entire process highlights how ketone bodies act as crucial energy sources during fasting and support lipid synthesis in early development. Succinyl-CoA serves as the source of CoA in this process. Beta-catechiolase then converts acetoacetyl-CoA into 2 molecules of acetyl-CoA. These acetyl-CoAs are what feed into the TCA cycle to produce energy. It's important to understand that the key enzymes involved in ketone body synthesis, HMG-CoA synthase and HMG-CoA lyase, are found in the liver and kidney cortex, but not in other tissues. On the other hand, the enzyme responsible for ketone body utilization, acetoacetate succinyl-CoA transferase, is present in many tissues, but notably absent in the liver. This distribution of enzymes ensures that the liver is the main site of ketone body production, while other tissues, like muscle and brain, are equipped to utilize these ketone bodies for energy. So, in essence, these differences in enzyme locations are what allow the body to efficiently produce and use ketone bodies as an alternative energy source during fasting or low carbohydrate conditions. Let's continue exploring ketone bodies and their role in our metabolism. As we saw earlier, ketone bodies are produced primarily when carbohydrate availability is low, such as during fasting or a ketogenic diet. They serve as an alternative energy source for many tissues, especially the brain, which normally relies heavily on glucose. It's important to highlight that the main ketone bodies, acetone, acetoacetate, and beta-hydroxybutyrate, are produced in the liver through a process called ketogenesis. This process becomes active when acetyl-CoA levels are high and oxaloacetate is depleted, shifting the liver's metabolism toward ketone production. Once produced, these ketone bodies are released into the bloodstream and transported to other tissues. These tissues, including muscle and the brain, can convert ketone bodies back into acetyl-CoA, which then enters the citric acid cycle to generate energy. This mechanism is crucial during periods of prolonged fasting or carbohydrate restriction because it helps maintain energy supply when glucose is scarce. It also underscores the body's remarkable ability to adapt its fuel sources based on nutritional availability, reinforcing the overall theme of metabolic flexibility and efficiency. This slide doesn't contain enough information. That's why we're moving to the next slide. This issue is particularly common in newborns and preterm infants. The reason is that their bodies may not synthesize enough carnitine or may lose it through the kidneys. When carnitine levels are low, it can lead to some noticeable symptoms. For example, hypoglycemia, which is low blood sugar, can occur because the body struggles to break down fats for energy. We might also see lipid accumulation, meaning fats build up in tissues where they shouldn't, and muscular weakness, because muscles rely on proper fat metabolism to function well. The good news is that treatment is relatively straightforward, mainly oral supplementation with carnitine to restore normal levels. Recognizing these clinical signs early is crucial, especially in vulnerable infants, to prevent more serious complications. This ties into our overall theme of understanding metabolic deficiencies and how targeted therapy can effectively address them. This slide discusses Refsum's disease, which is an autosomal recessive neurologic disorder related to lipid metabolism. Essentially, it's a genetic condition where the body has a defect in breaking down certain lipids, specifically phytanic acid and phytol. These are dietary lipids that you get from chlorophyll in plant foods. Because of this metabolic defect, these substances start accumulating in the body, leading to various neurological and visual issues. Individuals affected by Refsum's disease may experience retinitis pigmentosa, which affects their vision, often causing night blindness and peripheral vision loss. They might also have diminished deep tendon reflexes, indicating neurological involvement, and problems with coordination, which can impact daily activities. Understanding this disease helps us see how metabolic defects can have widespread effects, especially in neurological health, and emphasizes the importance of proper lipid metabolism in maintaining overall function. This ties into the broader theme of how genetic and metabolic disorders can influence multiple systems in the body. Today, I want to talk about a very rare inherited condition called Zellweger's syndrome. It occurs in individuals who completely lack peroxisomes in all their tissues, which is quite unusual. Because of this absence, these individuals can't carry out the normal functions of peroxisomes, leading to a buildup of certain fatty acids, specifically C26 to C38 polyenoic acids, in the brain tissue. This accumulation has a significant impact on brain development and function. In addition to the buildup of these fatty acids, there's a generalized loss of all peroxisomal functions, which affects many aspects of cellular health. The disease manifests with severe neurological symptoms, things like developmental delay, muscle weakness, and profound neurological impairment. Unfortunately, most patients with Zellweger's syndrome don't survive past the first year of life, highlighting how devastating this condition is. Overall, this syndrome underscores the critical role that peroxisomes play in maintaining normal cellular and neurological health, and it ties into our broader discussion of cellular organelles and their importance for human health. This slide explains how Jamaican vomiting sickness occurs. It's caused by eating the unripe fruit of the ackee tree, which contains a toxin called hypoglycin. This toxin plays a crucial role because it inactivates an enzyme called medium and short-chain acyl-CoA dehydrogenase. Now, this enzyme is essential for a process known as β-oxidation, which is how our bodies break down fatty acids to produce energy. When β-oxidation is inhibited, the body can't properly generate energy from fats, leading to hypoglycemia or low blood sugar. Interestingly, this condition is linked to a deficiency in mitochondrial medium-chain acyl-CoA dehydrogenase, or MCAD, which is an enzyme normally responsible for metabolizing medium-chain fatty acids. So, in essence, the ingestion of unripe ackee fruit disrupts normal energy production, resulting in the symptoms seen in Jamaican vomiting sickness. This highlights the importance of understanding how certain foods and toxins can impact metabolic pathways and lead to serious health issues. Now let's talk about ketonemia and ketonuria. When there's a higher than normal amount of ketone bodies in the blood, that's called ketonemia. If these ketones are present in the urine, it's known as ketonuria. When these conditions occur together, we refer to the overall state as ketosis. It's important to note that ketosis can be a normal response in certain situations, like during periods of high fat intake or after intense exercise, especially in the post-absorptive phase. These are non-pathologic or harmless forms of ketosis. However, when ketosis occurs in the context of diabetes mellitus, it can become a serious problem. This is where ketoacidosis comes into play, a condition we'll explore further. So, to sum up, ketosis involves elevated ketone levels, which can be either benign or harmful depending on the context, and understanding this distinction is crucial in managing metabolic health. Let's explore why the rate of ketogenesis is promoted in untreated diabetes mellitus patients. When insulin levels are low, as is the case in untreated diabetes, the body perceives this as a state of relative starvation. Because insulin normally inhibits lipolysis, its deficiency leads to an increase in the breakdown of triglycerides in adipose tissue. This results in a surge of free fatty acids entering the bloodstream. These free fatty acids are then taken up by the liver, where they are converted into ketone bodies through a process called ketogenesis. The reason this process is promoted is that, without sufficient insulin, the body shifts to alternative energy sources, mainly fats, since glucose uptake by cells is impaired. This shift is part of the body's effort to maintain energy supply to vital organs, especially the brain, which can use ketone bodies as an alternative fuel when glucose is scarce. However, this overproduction of ketone bodies can lead to ketosis, and in severe cases, diabetic ketoacidosis. So, in summary, the promotion of ketogenesis in untreated diabetes is primarily driven by low insulin levels, increased lipolysis, and the body's attempt to compensate for impaired glucose utilization by switching to fat as the primary energy source. In this session, we have been studying the metabolic pathway of unsaturated fatty acids, those with odd number of carbon atoms, which require modification of the beta-oxidation pathway. So, to summarize, the key points are that unsaturated fatty acids follow a somewhat different route compared to saturated ones, especially when they have odd numbers of carbons. We saw that these odd-chain fatty acids need special enzymes to handle their unique structure, particularly during beta-oxidation. This modification is crucial because it ensures complete breakdown and proper energy extraction from these molecules. Understanding these pathways helps us see the bigger picture of lipid metabolism and how our bodies efficiently utilize different types of fats. Overall, this highlights the importance of specific enzymatic steps in maintaining metabolic flexibility and energy homeostasis. And this knowledge ties back to our broader theme of metabolic diversity and adaptation in human biochemistry. In this study session, you have learned about several important aspects of fatty acid metabolism. First, we discussed the oxidation of unsaturated fatty acids, which involves specific enzymes to handle double bonds and ensure complete breakdown for energy. Then, we looked at how fatty acids with an odd number of carbons are oxidized differently, producing unique metabolites like propionyl-CoA that need to be processed further. We also explored alpha and omega oxidation pathways, which serve as alternative routes when the main beta-oxidation pathway is impaired or overloaded. Additionally, we examined how fatty acid oxidation is tightly regulated to maintain energy balance and prevent excessive accumulation of intermediates. Moving forward, we delved into ketone bodies, what they are, how they're produced during fasting or carbohydrate restriction, and their significance as an alternative energy source, especially for the brain. Finally, we summarized the key points, emphasizing how understanding these processes is crucial for grasping metabolic health and disease management. This comprehensive overview ties back to the overall theme of lipid metabolism, illustrating the complexity and elegance of how our bodies process fats efficiently and adaptively. Let's take a moment to reflect on some key concepts we've covered. First, what is Zellweger's syndrome? It's a rare genetic disorder characterized by a defect in peroxisome biogenesis, which impairs the breakdown of very long-chain fatty acids and other substances. This leads to a buildup that can cause serious neurological and developmental issues. Understanding this helps us appreciate the importance of proper peroxisomal function in lipid metabolism. Next, how are ketone bodies synthesized? During periods of fasting or low carbohydrate intake, the liver converts fatty acids into ketone bodies like acetoacetate and beta-hydroxybutyrate. This process provides an alternative energy source for tissues, such as the brain when glucose availability is limited. It's a vital adaptation that ensures energy supply during metabolic stress. Now, let's outline how fatty acid oxidation is regulated. The process is tightly controlled at multiple levels, including enzyme activity, like carnitine palmitoyltransferase I, and hormonal signals such as insulin and glucagon. These signals determine when fatty acids are transported into mitochondria and broken down for energy-maintaining metabolic balance. Regarding α-oxidation, this occurs specifically in the peroxisomes. It's crucial for degrading branched-chain fatty acids, like phytanic acid, that can't be processed by the usual beta-oxidation pathway. Finally, I encourage you to think about how these processes interconnect with an overall lipid metabolism. They highlight the body's remarkable ability to adapt and regulate energy production, especially under varying nutritional states. Keep these concepts in mind as they form the foundation for understanding metabolic disorders and their treatments.